Hello everyone, my name is Hao Qi, the research scientist of GemPharmatech. Today, I will share you some information about our multiple novel mouse models of Alzheimer's disease. So about Alzheimer's disease, this disease was discovered in 1901 when Dr. Alzheimer's examined and observed a patient. He used cell staining technology to identify amyloid plaques and the neuron fibrillary tangles within the cortical gray matter of the brain. And in 1906, he gave the presentation which is linked with these pathologic findings with the clinical symptoms of dementia. So Alzheimer's disease is a central neurodegenerative disease with some pathological features, including the A beta plaques and also the tangles and the shrunken brain. And given the data collected in World Alzheimer's Report in 2015, over 46 million people suffered dementia worldwide. This number is estimated to increase to 131 million by 2050. So the total estimated worldwide cost of dementia is a huge number, which is 818 billion in 2015, and it will reach 2 trillion by 2030. So when the disease worsens, the patients entirely rely on caregivers for daily trivias and even for basic personal care tasks, which imposes a serious social burden. And there are some hypotheses about Alzheimer's disease, including the abnormal deposition of amyloid beta. So for the A beta accumulation, I would like to introduce some background information about this APP gene. So APP is a precursor protein of A beta, and APP has both amyloidogenic and non-amyloidogenic pathways. So in non-amyloidogenic pathways, this APP protein is first cleaved by the alpha secretase, which formed the SAPP alpha protein. This SAPP alpha protein is reported to have biological functions in growth regulation and neuroprotection. And the amyloidogenic pathway, APP, is first cleaved by the beta secretase and then cleaved by the gamma secretase, which will form the A beta. And the 42 long peptide form of A beta is more likely to accumulate and form hard insoluble plaques. So the A beta 42, we can say, is more toxic. And some mutations related to A beta accumulation were found in familial AD patients. The most common mutation in familial Alzheimer's disease patient is Swedish mutation. And the Florida, London, and Indian mutations in this APP gene would increase the ratio of A beta 42 to A beta 4T, which means these mutations will increase the toxicity of A beta. And this slide shows AD candidate drugs development status. So now more than 280 drugs are in the clinical stage. They mainly target A beta, Tau, BSAE1, and gamma secretase, and so on. So, however, few drugs make it to phase 3 or move into market. This suggests that we require more suitable preclinical model for drug evaluation. And animal models of AD have become valuable tools to understand familial or early onset AD. There are several conventional AD models, such as 3TG, 5FAD, and the APP-PSEN1 based model. So about APP-PSEN1 mice, these mice began to develop a beta deposition at 6 months of age, and spatial learning defects by 12 months. And for 3TG mice, because these mice carry the mutations related to Tor protein, which is the MAPT P301L mutation. So we can observe the neuron triangles in these 3TG mice at 12 months. And about 5FAD mice, these mice express human APP and PSEN1 transgenes with a total of 5 AD-linked mutations, and that's why this model called 5FAD. So the mutations include the Swedish mutation, the Florida mutation, and the London mutation, and other 2 mutations in PSEN1 gene. And this 5FAD model, the A-beta deposition occurred much earlier than other 2 mice models. And also the spatial learning defects could be detected by 4 to 5 months. So, in order to better understand disease pathophysiology, as well as to search for models suitable for investigating potential therapeutics, we established FAD4T mice. So what is FAD4T mice? These mice have 4 AD-linked mutations, the Swedish and Indiana mutations in APPG, and the other 2 mutations in PSEN1 sorry. and compared with 3TG and 5FAD mice, which were not available for companies or for commercial use, our FAD4T mice have independent IP, and the models are available for all customers. And amyloid plaques are observed in mice as young as 1.5 months of age. And amyloid pathology is more severe in females than males, which is consistent with the clinical. So next I will show some data of FAD4T mice. By using the antibody that specifically reacts with A-beta plaques, we detected A-beta deposition as early as at 1.5 months. And A-beta deposition increased gradually with age. And the A-beta plaques is more severe in females than in males, and this part of data I didn't include in this PPT file. And for the A-beta deposition, this is the data from one of our collaborators, and the A-beta deposition in 4-month-old FAD4T mice are comparable to 5FAD mice, both in cortex and in hippocampus. And astrocyte activation is another important marker of AD. So we detected astrocyte activation by GFAB in FAD4T mice at different ages. And significant glial hyperplasia was observed in FAD4T mice at 7 months of age. So to evaluate the spatial learning and memory ability of our FAD4T mice, we perform the classic Morris automate test. So I would like to introduce some details of how we perform this Morris automate test. So the maze was equally divided into 4 logical quadrants and a hidden platform. The training session lasted for 4 consecutive days and 4 trials per day, and was followed by a prop test on the 5th day. In each trial of training session, mice were randomly released into the water from 4 quadrants of the pool and allowed to have 90 seconds for finding the hidden platform and 30 seconds to rest on the platform. So, about the results, for 3-month-old mice, the FAD4T mice showed normal learning and memory abilities compared with wild-type mice, which is shown in Figure A and Figure B. While in the spatial search experiment, compared with wild-type mice, there is a tendency of cognitive decline in 5 FAD4T female mice, indicated by less transition number, the number of crossing platform, and spent less time in the target quadrants here. So to further confirm the when we can detect the spatial learning and memory defects in our FAD4T mice, we collaborated with one of our customer. So in this result, the mice were subjected into learning paradigm of auto-based test for 6 days, which is different from the previous experiment, and followed by a probe trial at day 7. So in each trial of training session, mice were randomly released into the water from 2 quadrants of the pool and allowed to have 60 seconds for finding the hidden platform and 15 seconds to rest on the platform. As shown in the learning sessions, which are Figure A and Figure B, the female and male FAD4T mice displayed a significantly longer latency, indicating the FAD4T mice were impaired in their spatial learning abilities. And in the prop test, the number of crossing platform was decreased significantly in female FAD4T mice, which indicates defects in spatial memory in the FAD4T female mice. So these above results indicate that 5-month-old FAD4T mice have spatial learning and memory impairments. We also did efficacy evaluation with our FAD4T mice. This is a case to show that our FAD4T mice is suitable for efficacy test. So we first trained different groups of mice for 5 days, which include the control, the wild-type group, the FAD4T mice without any treatment, the FAD4T mice with donepezil treatment, and also 3 doses of the drugs from our customer. So as we can see, during the training, our FAD4T mice need more time to find the platform and had significant defects in spatial learning compared with wild-type mice, and the treatment of low dose of drugs could rescue these defects. And the results of probe test further confirmed this. the number of crossing platforms was first significantly decreased in our FAD4T mice compared with wild type, and then it was rescued by the treatment of low dose drug, which is consistent with the learning sessions. And also the time spent in the target quadrants was possess similar results. And also the time in the platforms, our FAD4T mice significantly spent less time in the platform, and with the treatment of low dose drug, the time in the platform was increased. So our FAD4T mice also shows spatial working defects in spatial working memory in Y-maze test. As you can see, the alternation of female FAD4T mice is significantly decreased compared with wild type, and also the number of arms entered. And in the open field test, we did not observe differences between wild type and FAD4T mice in locomotor activity. So those differences in the Morris automate and the Y-maze test were not due to the locomotor activity. So in a summary, a beta deposition could be detected at 1.5 months in our FAD4T mice, and for now we didn't observe the neuron triangles in our FAD4T mice. And about the behavior defects, we observed spatial learning defects by 6 to 8 months. So these features make FAD4T mice a very useful model to study AD and evaluate the efficacy of AD drugs, especially targeting A beta. And other than this FAD4T mice, we are also working on the next generation of AD mice models, which we call it AD plus model. So what is AD plus model? First, beside A beta accumulation, there are some other possible causes of Alzheimer's disease, such as inflammation, metabolic disorders, and infections. And more than 20 genes are very highly associated with Alzheimer's risk, such as ApoE and TRIM2, and also the PSALM and some other risky genes. So based on this, we are now developing AD plus models. We first generate humanized risk genes of AD and then cross these mice with our AD model to make AD plus models. So if you are interested in some of the risky genes of AD, you can use our AD plus model, because this AD plus model have the features of Alzheimer's disease and also have the humanized target you are interested in. And here is a list of AD plus models. So most of these models will be available in this year. And those highlighted ones are very popular according to the feedback from our department of market colleagues. So what's more, we are also developing some very normal mice for Alzheimer's disease. First, I would like to introduce some background information about this chromosomic stress. So we introduced the chromosomes from wild mice, which were captured in the wild, into our B6 mice, and because there are some disadvantages of the inbred mice we are now using for our experiments and studies, because the genes variety is much limited in the inbred mice. So to solve this problem, we introduced the chromosome of wild mice into the inbred mice to generate the chromosomic stress. So after around 6 to 7 rounds of cross, we generated the chromosomic stress, and we now have a theory of chromosomic stress with chromosome 1 from the wild mice. We named one of the chromosomic strands as 765, and this 765 mice has the chromosome 1 from the wild mice, and the other 19 chromosomes were chromosomes of B6. So, interestingly, we observed these mice had better spatial linear ability according to the results of Morris Watermace. As shown here, both the male ones and the female ones, this 765 mice had decreased latency of platform, so these mice could find the platform earlier than the wild-type B6 mice. And in the probe test, the platform location crosses the male 675 mice crossed much more than the male wild-type B6. And also for the time spent in the quadrants, the male 765 mice spent much more time in the targeted quadrants than in wild-type B6. And the speed of these mice were similar to the wild-type B6. And what's more, we further crossed this 765 mice with our with AD model, which is the 5F80 mice. And this research was conducted with one of our collaborators. And surprisingly, the 765 crossed with 5F80 mice developed more pronounced spatial memory defects, which is opposite to the previous result. So although these mice had a better spatial learning and memory ability, when these mice was crossed with AD mice, the phenotypes of AD were much more obvious and severe. And about the A-beta deposition, consistently this 765 mice crossed with 5F80 mice displayed more amyloid deposition with decreasing age, both at 3 months old and 6 months old. So although we haven't figured out what's why this 765 mice is so much different with the wild-type B6, but hopefully we can find out some novel and interesting targets, both for the spatial memory learning and memory, and also the Alzheimer's disease. So other than Alzheimer's disease, we are also working on other neurodegenerative diseases, including Parkinson's disease, ALS, Huntington's disease, and those started ones are what we are working on. And here is a list of part of our models, including the targets and estimated time to step into the market. So the PD, ALS, and HD models will be available for collaborative validation by the end of this year, and available in the first or to the second quarter of 2022. And the SMA model is scheduled to be available in the third quarter of 2003. And if you are interested in our models, you can contact us at any time and discuss those cooperation matters. And that's all. Thank you. Thanks for your attention.